Boa tarde, pessoal. A professora Andréa vai entrar daqui a pouquinho. Nós vamos começar, então, mais um seminário da nossa série. So it's my pleasure to introduce today's speaker, Dr. Karthik Anantharaman. He is assistant professor at the Department of Bacteriology at the University of Wisconsin, US. He received his PhD from the University of Michigan in 2014, studying deep sea hydrothermal vent microbiology using multiomics approaches. His interdisciplinary research focuses on understanding the cycling of sulfur and other nutrients, with a strong emphasis in microbial and viral processes that transform these cycles in the environment and human systems. His lab is at the forefront of developing bioinformatics and statistical approaches to investigate microbiomes, viromes, metabolism, and community interactions. And today he is going to talk to us about systems biology of virus in human and environmental microbiology. And with that, I would like to pass the word to Dr. Karthik. So Karthik, we are all yours. You can share your screen.
Thank you so much, Dr. Kruger, for such a fantastic introduction. And thank you so much for the opportunity and the invitation to present.
It's a pleasure. I will stop my video and mute my audio. So,
can you all see my screen? Okay, thank you so much. So my name is Karthik Anantharaman. As Dr. Kruger introduced, as you saw from Dr. Kruger's introduction, I am an assistant professor at the University of Wisconsin-Madison. My laboratory is a very multidisciplinary laboratory in a way where we study a variety of human and environmental microbiomes. And that's the sense that I will hopefully convey today in my talk. My laboratory is somewhere at the confluence of three different fields. We work in chemistry, primarily geochemistry and biogeochemistry. We conduct research in microbial and viral systems ecology, and finally computational biology. So I like to think, we are somewhere in the center of all of these fields. The primary question that my laboratory asks is trying to understand the role of microbes and viruses in driving sulfur metabolism, human health, and disease. During my PhD and my postdoc, I studied a lot of environmental microbiology. I studied biogeochemistry and sulfur transformations, and I've tried to take a lot of that into my own independent lab today, but I've also diversified into studying sulfur metabolism in human systems and how sulfur metabolism can actually impact human diseases. When you think of our lab, we have three different research themes in the lab. We study deep sea microbiology, so we study hydrothermal vents and the deep oceans. We primarily study inorganic sulfur transformation. So hydrothermal vents or deep sea volcanoes are very rich in sulfur. So we try and study the microorganisms and viruses that are transforming this inorganic sulfur at hydrothermal vents. We study freshwater microbiology. So we study both tropical as well as temperate lakes. So lakes here in Wisconsin are temperate lakes, but we also have projects in Africa looking at tropical lakes. And the key here is, again, looking at inorganic sulfur transformations. Microorganisms that are present in lakes can really affect the productivity of the lakes. And as the earth warms due to climate change, it's predicted that lakes will become more stratified. So sulfur is going to play a critical role in trying to understand in the future of lakes. And finally, our lab studies organosulfur or organic sulfur transformations in the human gut. We are primarily right now interested in IBD, which is intestinal bowel diseases, as well as colorectal cancer. Now, in addition to studying sulfur transformations, we also develop software tools to study microbiomes and viromes. So three softwares that my lab has developed right now include Vibrant, which stands for virus identification by iterative annotation. And this Vibrant extracts viral sequences from different kinds of DNA. Primarily, this was designed for metagenomics data, but it can be used with any sort of genomics or single cell genomics data as well. We have another tool called Propagate, and this one is also designed for viruses. So this one specifically looks at prophages, which are phages or viruses that integrate into host genomes, and it identifies whether these prophages are active. So that's what Propagate stands for. And finally, we have one called Metabolic, which enables metabolic and biogeochemistry analyses in microbes. Again, Metabolic is designed primarily for metagenomics data, but can be really used with any sort of genome data. So in essence, today I'll try and give you a sense of the different kinds of questions that you can ask with some of these tools, specifically Vibrant and Propagate, because I'll try and focus this talk on viruses. But I'll also give you a sense of our work in the deep sea. So, first we'll talk about bioinformatics, a little bit of work in our human microbiome space, and then we'll switch gears and talk about our research in the deep oceans. So we are an actively field-going lab. So we conduct research in the field. We go on research cruises, so ships in the ocean, and we have used submarines such as Alvin to go down to 2,000, 3,000 meters below the surface and actually collect samples from hydrothermal vents. So we're an active seagoing lab. I'll show you a lot of interesting and fun pictures and videos as well from the field so that you can appreciate what it's like to look at hydrothermal vents, look at sulfur processes and microbes and microbial mats, and how difficult and challenging it can be as well. So let me start off with an introduction, a very brief introduction that is about viruses. When you think of viruses, viruses really are the most abundant biological entities in virtually every environment. They are very abundant. They control the availability of nutrients. They can kill bacteria as well as other organisms, and in doing so, they influence microbial community composition and diversity. I'm just going to pause briefly and try and use a laser pointer. Okay, so you can see okay, so I'll use the laser pointer to just highlight where I'm at. But so viruses can kill bacteria and other organisms, and in doing so, they influence microbial community composition and diversity. And this is not something we often associate with viruses, but viruses, in fact, can drive metabolic processes in the environment. So I'll sort of discuss, different ways in which this is done. But when you think of, say, what viruses look like, viruses these are, traditional shapes or ways in which bacteriophages or phages that infect bacteria look. But viruses have different kinds of shapes and forms. And finally, when you think of, say, viruses are really, really small. So in the context of abundance, we all often see that viruses are extremely abundant. So for example, in the oceans, viruses can constitute up to 95% of all particles or entities in the ocean. But if you were to look at the biomass associated with viruses, it would be a very small fraction. So 95% of all particles are viruses, but only 5% of all biomass in the oceans actually comes from viruses. So even though they're small, they actually play a very large role in ecological processes. Now, what is the life cycle of a virus? Or what we're going to look at is just the life cycle of phages. The way phages that infect bacteria work is they can land on a cell and inject their DNA into the cell. Once the DNA is injected, this DNA is copied. So you have multiple copies of this DNA forming. It starts off with some form of gene expression, host modulation, followed by propagation of this DNA. After this, virions or virus particles are assembled. And once there is a critical mass of these virus particles, the cell lyses and the virus particles are released into the environment. And this is a very traditional sort of a life cycle of most viruses, including coronavirus or COVID-19, very similar. We can also study these processes in the laboratory. So our lab, we also conduct wet lab experiments, some of which I will show today. But in essence, what you're looking at over here on the extreme right is a thick culture that contains microorganisms or bacteria. And then once you infect this bacterium with a virus, slowly over time, you will see that the solution clears. So essentially, this virus has killed the bacterium, and that's what you see by the solution clearing. Now, why should we study viruses? Now, in the gut, for example, there are a lot of unknowns with regard to viruses. Now, viruses, along with bacteria and some protists, constitute the microbiome. Now, this microbiome interacts with the host genome to generate immune responses, which can either protect us against diseases, or it can also exacerbate diseases, or say increase the potential of diseases. But what is important is that we know next to nothing about the role of bacteriophages in the etiology of most diseases associated with the gut microbiota. We've sort of figured out that microorganisms, so specifically bacteria, are responsible for a lot of diseases, but we don't know the role of viruses. And that is something as a lab we are trying to change. Now, viruses can also manipulate microbes through a process known as viral auxiliary metabolism. What is auxiliary metabolism? This is the idea that viruses can often steal genes from their hosts. And then when they infect that host, they can use those genes to manipulate the host. So under a normal scenario, you would have a virus that infects a bacterium, it'll kill the bacterium, and then the viruses would escape. So that's what's shown in this fun image over here. But if viruses were to undertake auxiliary metabolism, rather than killing their hosts, viruses would infect the host and increase the life cycle of the host. When the host is alive, the virus is forcing the host to conduct a certain process and then utilizing some energy that is produced by the host to produce more virus particles, but the host itself does not die. So this, in a way, somewhat benefits the host, but it also benefits the virus a lot. And that's the concept of viral auxiliary metabolism. Now, this is something our lab studies a lot. And we are currently studying this particular process both in human microbiomes and in environmental microbiomes. And I'll show you some examples associated with this today. Now, I mentioned that some of the primary questions that we seek to ask in our laboratory are trying to understand the roles of virus-driven metabolism in modulating microbiomes and health. Now, we ask this particular question along 3 different lines. So first, we try and understand the diversity of viruses that exist in a system. So a question that we can ask is, how can we accurately identify and characterize viruses and viral dark matter from microbiome data? So if you're wondering what viral dark matter means, viral dark matter is just the uncultivated viruses and proteins that are present on those viruses, or say encoded by those viruses. These are commonly referred to as dark matter. We are also interested in the distribution of viruses, essentially trying to ask the question, can we use viruses as predictive markers of disease? And finally, we are interested in ecology. So we try and ask questions such as, what are the roles of viruses in driving dysbiosis? And dysbiosis is the idea that the gut microbiome is in a dysbiotic state, which means there's been some issue with the gut microbiome which might drive disease. So we try and ask questions along these lines. Now, what I'm going to show you here is the first bit of it. So I'll show you some bioinformatic tools that we have developed trying to ask the question, how can we accurately identify and characterize viruses from microbiome data? Now, much of what you're going to see here is research that is driven by Christopher Kieft, who is a 4th-year PhD student in my laboratory. Now, as far as microbial and viral ecology goes, I like to think we take what I call a top-down approach to tracking microbial and viral ecology. Now, this can be any system, okay? It can be human gut, so it can be stool samples, or it can be filters that come from the deep sea. But our first step starts off with extracting DNA and RNA and then conducting shotgun sequencing. So we typically, try and look at the identity of organisms. So try and ask who is there. Try and look at the metabolic potential. So what are the organisms, be it microbes or viruses, capable of doing? And then finally, look at growth rate. So are the organisms growing? And if they're growing, how fast are they growing? Our research is typically genome resolved. So, from shotgun sequencing, we generate organisms through a variety of bioinformatic approaches, and we get different genomes that would look like this. We reconstruct metabolisms. And then finally, we undertake ecological reconstructions that look like this. Now, when it comes to studying viruses, viruses are slightly trickier to study than bacteria. And the reason is that we don't have a good idea for the most part of what viral genomes look like. So we have to we have to develop new tools or come up with newer approaches to try and study viral genomes. One of those approaches involves using machine learning. So we are currently using machine learning approaches to differentiate between microbes and viruses, essentially differentiate between microbial and viral genomes. What you're looking at are DNA sequences from a variety of organisms. So you have DNA sequences from bacteria and archaea in blue, you have plasmids in red, and you have virus sequences in green. And essentially what this is showing is that we have a really good machine learning model that can separate viral genome sequences over here from bacterial and plasmid sequences. Now, this machine learning model is implemented in a software tool known as Vibrant. Vibrant can be used as a standalone software on your computer or your server, or you can also use it on the web server Cybers. Vibrant, the manuscript itself is published, so I welcome you all to check it out. But how does Vibrant really work and what can it do? So Vibrant identifies viral signatures, and it uses three different databases as its three different databases and the annotations present in it into its model. Essentially, what it's doing is it identifies the proteins present on different sequences and then tries to identify whether those proteins are likely to belong to a bacterium or a virus. So there are some statistical analyses over here. The model itself is a supervised machine learning model, and we have validated cutoffs. So these, this model is the reason that Vibrant can achieve better efficiency than a lot of similar programs. Now, this is about how Vibrant works, but what can Vibrant do? Vibrant can identify phage from metagenome data. It can also identify phage that are present in single cell data or other kinds of genomic data, like say from isolate genomes as well. It can annotate phage genomes, and it can also determine genome quality and say completeness as well. And the completeness is sort of qualitative, but it is still a measure of completeness. After this, it can identify genes and metabolic pathways that may be present in the phage genomes. And then finally, it can help visualize your data. Now, I'm not going to show a lot of different kinds of visualizations, but I will show this. So Vibrant, for example, can help you understand how viruses can impact metabolism in specific bacteria or the environment. So what you're looking at over here is an example environment. So this is data that comes from the ocean, so marine systems. And what this is showing you is the viral community and the metabolic genes that are present on the viral genomes that came from this marine system. So for example, what this is showing you is that viruses in marine systems are more likely to have genes associated with energy metabolism than carbohydrate metabolism. Or for example, viruses are more likely to have genes associated with amino acid metabolism than say nucleotides. So this is something that you can then follow up with regard to specific wet lab experiments or other kinds of experiments if you're interested in viruses. Now I'll show you certain, so I'll show you how we validated Vibrant, but also this will also sort of give you a sense of the different kinds of questions that you can ask with Vibrant. Now, to validate the performance of Vibrant in the human gut, we tested it on a specific Crohn's disease cohort. So this particular cohort of Crohn's disease patients had 59 individuals in the diseased cohort, and there were 37 healthy controls. We had metagenome data from this particular cohort of 96 individuals, and in total we identified 14,000 viruses. Now we wanted to ask different sorts of questions associated with this. The first question we asked was, are there viruses that are unique to Crohn's disease? That is, viruses that would be present in Crohn's disease, but not in any of the healthy controls. So we conducted a lot of analyses of shared viral genomes as well as shared genes between the viruses to identify this. Next, we asked if viruses are differentially abundant between Crohn's disease and controls. And here we conducted differential abundance analyses of viruses that are shared between both data sets. After this, we tried to ask if there are signatures associated with replication or production rates of viruses between Crohn's disease and control. So essentially what this is asking is, if there is a virus that is present in a healthy gut and in the gut of a patient with Crohn's disease, are the replication rates of the viruses the exact same, or are certain viruses growing faster in Crohn's disease? So we conducted differential replication index analyses of viruses that are shared between both these data sets. Now, I'm not going to show you all of the analyses here, but I will sum it up. But what I will show you specifically are viruses that are differentially abundant between Crohn's disease and controls. So the first question is, are viruses differentially abundant? The answer is yes, there are shared viruses between these two diseases that are differentially abundant. What you're seeing over here are just examples of two specific viruses. So here is one virus, virus 1, that contains 2 interesting metabolic genes, and then virus 2 that contains 3 interesting metabolic genes. If you look at the abundance of these viruses, that's what the y-axis shows. In healthy patients, you see that the virus is present at low abundance, whereas in Crohn's disease, it's typically present at higher abundance. Very similarly, this second virus is also present at higher abundance in Crohn's disease and present at far lower abundance in healthy patients. And in both cases, these analyses are all statistically significant. So to summarize what we saw with regard to viruses in Crohn's disease, the first question was, are there viruses that are unique to Crohn's disease? And the answer was yes, there are viruses, and these are viruses that infect Fusobacteria and Enterobacteria. Second, are there viruses that are differentially abundant between Crohn's disease and controls? The answer is yes, there are viruses that are differentially abundant, and it seems like these are Enterobacteria viruses. Third question was, are there signatures associated with replication rates of viruses between Crohn's disease and controls? And that's what I just showed you. The answer is yes, there are viruses with very specific metabolic genes associated with toxins that have differential replication rates between the two states. Now that's it about Vibrant, but I would also like to show you some data associated with a brand new software tool that we have just released, and just sort of give you a sense of the capability of this particular tool. This tool is known as Propagate. Propagate is a prophage activity estimator. So that's what the logo also is trying to show you. If you imagine this is a genome and this is a integrated virus, so this is a prophage, then at some point of time, this prophage will get out, and that's what we are trying to identify. So it identifies prophages that are actively replicating. If you think of the different stages of infection by prophages specifically, so prophage in a dormant stage, you have this prophage here in green that is integrated into the host genome. So this can be bacteria or archaea. In an early lytic stage, so this is when the prophage activates, you will see that the prophage genome is expressed. So you have a copy of the prophage genome here separately, and it's also integrated into the host genome. In a mid-lytic stage, this is when the virions or virus particles assemble. You have three stages here. So you have the genome that could exist independently, and then the phage genome will also exist inside a virion. And then finally, you have the late lytic stage, which is the lysis stage. And this will have genomes in 4 stages. There'll be a genome that will be on the host genome, a prophage genome that will be independent, prophage genomes that will be inside viruses, but the virus itself is inside the bacterial cell. And then the bacterial cell is lysing. So there's also viruses that are escaping the bacterial cell. So 4 different stages here. Now, when the prophage is active, you'll have a mix of all of these stages. And that's essentially what we are trying to track. In an active prophage stage, typically you'll have cells that are a mix of at least these 2 stages for the most part. And in essence, what you will see, if you were to measure the total number of copies of this phage genome, you will see that they are much larger than the host genome. So for example, what you're seeing over here are 5 cells, but we can only measure host genomes in 4 cells. In this cell, it's sort of lysed, it's dying. So this has lost its genome, but there are 4 copies of the host genome that we can measure, but we can measure 23 copies of the prophage genome. So we measure the copies of the prophage genome and its ratio to the host. And that's essentially what tells how we identify activity of the prophage. Now, in contrast, if you were to look at a dormant prophage, a prophage that is not active, then you will see that they are present in about 1:1 copy. Okay, 1:1. It's never 1:1. It's very close to 1:1. And the reason is that some cells can often kick out the phage genome from their genome. So it's an evolutionary signal there, or it's an evolutionary process where certain hosts can throw out the phage genome. Now, how do we actually do this? So we do this through a process of mapping a read coverage or looking at read coverage patterns. The idea is if you were to look at read coverage for a genome, so this is essentially looking at how abundant a genome is. And let's assume this is the host and the prophage genome is here in green. So it has integrated into the host genome sequence. And if you were to just look at a dormant prophage, this is what you would see. The coverage would be very similar across the host and the prophage. But if the prophage were active, we saw there was about a 6:1 ratio I showed in the previous slide. So if you see something like that, then you will see something like this, where in the host ratio, the read coverage would be about certain number. And with regard to when the prophage is active, you will see a much increased read coverage in the prophage region. This is how you know that the prophage is active. Now, you can use this for a variety of different questions, primarily looking at activity of phages. So we've tested this in a number of environments. We've used this to study prophage activity, obviously using it right now to study prophage activity in the human gut, as well as in the environment in lakes and in hydrothermal vents. Here's just one example that we used in the environment. What you're looking at are Myxococcales, which is a particular group of bacteria. And you're looking at prophages here in red and the host genomes, or say host in blue. And the y-axis, so these are all different samples. So we looked at the prophages across different samples. And the idea here is that if you had spatial association, or say even temporal, so time or space, if you want to look at these, you can see how a particular prophage is being active. So for example, here we have a lot of different kinds of samples, and we're looking at the exact same host phage system. There's only one host and one prophage. So in this particular sample, you can see that the prophage is not very active. But in this sample, the prophage is extremely active. So clearly different chemistries or just different spatial structures and arrangements have caused the prophage to be very abundant in one sample versus another. So this is an environmental sample. This is not a gut sample, but I think this gives you a sense of how we can use propagate to look at differential activity of specific prophages. So that's all I'm going to talk about with regard to bioinformatic tools today. But the next few slides, I'll give you a sense of the different kinds of questions we are trying to ask with regard to understanding the roles of viruses in driving sulfur transformations. Now, specifically, we are studying human microbiomes that are associated with colorectal cancer and IBD. And this research, much of it is actually driven by an MD PhD student, Elise Cowley. So she is a clinician. She's a clinician. So she comes to us with a keen understanding of diseases and one is looking at how viruses as well as microorganisms are actually driving a lot of these processes in the gut. In addition to Elise, Christopher Kieft, my PhD student, is also working on these questions. And the key thing that they found is that there is an enormous diversity of sulfur genes that are encoded by viruses in human systems. Traditionally, we only studied the inorganic sulfur processes, such as how sulfate is reduced to certain intermediates, then to sulfite, and then reduced down to hydrogen sulfide. But in reality, what we have discovered is that there is an enormous diversity of sulfur transformations associated with a variety of inorganic, what you see over here, as well as organic compounds such as cysteine and methionine, other derivatives of amino acids like taurine. So there is a lot of different kinds of sulfur transformations that are taking place. And the key thing is that viruses have the genes to transform these sulfur compounds. Different viruses that infect different microorganisms contain different genes, but it is absolutely key that all these genes are very abundant. As an example, our laboratory is very interested in the degradation of cysteine to hydrogen sulfide. And the reason for this is that hydrogen sulfide is genotoxic. It's genotoxic in all environments, both in the human gut as well as in the environment. But in the human gut specifically, H2S can drive colorectal cancer, and it's also important for intestinal bowel disorders and Crohn's disease. Cysteine can be broken down to hydrogen sulfide through 5 different genes, and viruses, we have identified viruses that can carry that have each of these 5 genes. So different viruses have different genes, but yes, all the 5 genes have been identified on viruses, and we even have model systems where we are trying to study how viruses are utilizing these genes. These viral genes for the degradation of organic sulfur and production of H2S are quite abundant. Depending on the microbiome, you will see different sorts of genes. So for example, in oral microbiomes, we see that these genes are quite abundant, and in the gut as well, they're important, but they're not as abundant in the vaginal microbiome. But if you look at, say, these 3 pie charts, you can see different colors. So the take-home message really here is that the composition of genes associated with sulfur transformations is very different in oral systems versus the gut systems versus the vagina. Now, if you're wondering, why would viruses carry genes for production of hydrogen sulfide and sulfur transformations? The answer is that hydrogen sulfide seems to increase viral fitness. How does this happen? We don't exactly understand how or why this happens, but what we think is, but we have some hypotheses. What we can see is that as you increase sulfide concentration, the number of viral particles produced is greater. So more viruses are produced under higher H2S concentrations. So on the y-axis, you have %PFUs, or it's a measure of viral particles. And on the x-axis, you have H2S concentrations. So here's a control, and then you're increasing H2S concentrations like this. And you see that the number of viral particles increases, pretty much linearly. Now, these concentrations do not affect the microbial host. So one hypothesis we had was, is the microbial host affected? And is the pattern we are seeing, does it have something to do with that? And the answer is no. The microbial hosts are not affected. So clearly, the viruses are getting some sort of a benefit from H2S, but we are not able to understand what. So we have a particular model of virus-enhanced sulfide production in the human gut. So the idea is that there are genes for cysteine degradation that are present in viruses. This leads to uninhibited cysteine degradation in infected host cells, which eventually leads to enhanced viral replication. To just sort of show this visually, imagine that bacteria have abundant intercellular cysteine. So cysteine concentrations are very high in bacterial cells. You have a phage that infects this particular bacterium. And during the infection, the viruses are expressing CISK, which is a particular gene for degradation of cysteine. It's actually a multifunctional gene, but one of the processes that this gene catalyzes is degradation of cysteine. Bacteria can upregulate their cysteine degradation metabolism to produce H2S. And finally, viruses replicate inside the host cell. So this is sort of the process that we think is happening. Now, the H2S that is formed by the viruses can have a lot of other repercussions. So it can also cause increased intestinal inflammation. We don't know the extent of the H2S that's produced by the viruses and to what extent it causes intestinal inflammation, but we certainly think that there is increased intestinal inflammation that can be attributed to viruses. So to just think of, say, how this would look like. So if you had a healthy gut, you have your mucosal surface here, and then you have different bacteria, commensal as well as a pathogenic bacteria over here. If you had an infection by certain pathogenic bacteria, and let's say bacteria produce H2S, you would have some intestinal inflammation that looks like this. And if you had viruses as well, and both bacteria and viruses were producing H2S, then you would have increased intestinal inflammation that's coming from both sets, both bacteria as well as viruses. So I hope you can now appreciate how viruses can produce H2S as well as contribute to processes in the human gut, primarily associated with disease. But for the remaining time, what I'll do is I'll sort of change gears. And now I will talk about microbes and viruses in deep sea hydrothermal vents. And again, to remind you, if you're wondering, what's the connection between the human gut and hydrothermal vents of the deep sea, the clear connection that we look at is associated with sulfur. At the end of the day, microorganisms transform sulfur in certain ways, and that's what really drives the questions that we ask. Now, why should we study hydrothermal vents? Now, hydrothermal vents, as I mentioned, are a very important source of sulfur, but they really act as an interface between the lithosphere and the oceans. There's all sorts of chemicals that are coming from the Earth's crust. Now, these are commonly sulfur, iron, manganese. And I mentioned these three because these three are very important for the global oceans. For the oceans to remain healthy, microorganisms are really important, and microorganisms really need sulfur as well as trace metals, primarily iron. Iron is really important for photosynthesis. But in addition to these, there are a number of trace metals as well that come from hydrothermal vents. What you're seeing over here is iron concentrations. The hot colors or warm colors, so red, for example, indicates high iron concentrations, and green indicates slightly lower concentrations. But the key thing to look at over here are that so what you're looking at over here are longitudes. So you have a hydrothermal vent over here and you have longitude. So in essence, when you have iron produced by a hydrothermal vent, it can travel thousands of kilometers into the ocean. And this ion is also, complex to sulfur as well as other elements. So in essence, when you produce, with regard to hydrothermal vents, they can affect ocean chemistry on global scales. And that's similar to what you're seeing over here as well. Wherever there are hydrothermal vents, you will see elevated or say high concentrations of sulfur, iron, manganese, and a number of other trace metals. These hydrothermal vents are globally distributed. So all of these black lines that you see are mid-ocean ridges. And I've indicated the known and predicted hydrothermal vents in red and yellow. Now we have a number of field sites. Our lab has field sites in Lau Basin. So this is near Fiji in East Pacific Rise, so which is off the coast of Costa Rica here. And finally, Guaymas Basin. So this is in the Baja Peninsula right here. All the three systems are very different, but the key is that all of them are very rich in sulfur. So we try and understand how viruses are interacting with sulfur and its association with other elements in these three different environments. Now, what do hydrothermal vents look like? I thought I would show this particular video. And this has some, this is from a recent cruise I did about 2 years ago. So this was in 2019, where we had 4K cameras. So very high definition footage. So what you're looking at are hydrothermal vents. All of the water is at about 300 degrees Celsius. All the colors that you see are life, higher organisms, so eukaryotes, primarily ciliates, but there are also some worms and the different other colors. So there is also a lot of microbial mats. So the white and the yellow and the orange are all microbial mats. Because it's white and orange, we also call these fried egg mats. But there are worms over here. So there are tube worms. It's a beautiful tube worms. In purple are ciliates. But it is a really beautiful experience to try and say, go down in a submarine, for example, and look at these very close to you. It's a life-changing experience. But I'll show you some images associated with how we collect these samples as well. So if you can see, there's shimmering water here. So when this 300 degrees Celsius water hits the cold ocean, that's when you see that shimmering light. All of the white mats over here are bacteria. There's also bacteria in a number of the fluids around this hydrothermal vent as well. You saw the animals. There are symbiotic bacteria inside the animals as well. Yeah, so there are symbiotic bacteria inside of these tube worms as well. So this particular video was on a cruise that was supported by the Schmidt Ocean Institute. So we have a number of videos just like this. I'd be happy to send you links if you're interested in watching more of those. But an important reason why we study these specific environments and these sulfur transformations specifically here is that the kinds of processes that you see in hydrothermal vents also occur at other environments such as whale falls, or where you see large animals dying at say gas hydrates. So these are known as cold seeps, where you have a lot of methane and a lot of sulfur. Oxygen minimum zones. So these are the large zones around the ocean today where there is no oxygen. It's assumed that these cover about 5 to 10% by volume of the oceans. In coastal hypoxia, in eutrophication, and in productive surface waters as well. So all these processes can happen all across the ocean. Now, how do you sample a hydrothermal vent at the bottom of the ocean? We use a number of in situ filtration processes. So we essentially filter underwater. So what you're looking at over here is a process known as Super, or say instrument known as Super. So this is a sampler that can be loaded onto a remotely operated vehicle. So this is a robot. This Jason that you see is a robot. You can load Super on here, and then when it goes down, you can sample. Super has about 24 ports, so each of these is one filter. So what you're seeing is, we have filtered 24 different samples, and these samples contain microbes as well as viruses. In addition to this, we've also designed our own filtration equipment known as Hi-Fi, which stands for high throughput inline filtration of viruses. Now, when we are sampling them, we load our instruments on Super, or say on a robot, such as ROV Jason. We send ROV Jason down, and then we can sample any point of the hydrothermal vent or the hydrothermal vent fluid. So we can sample here, here, and this is typically what we do. We sample very close to the hydrothermal vent as well as the deep sea, as sulfur transformations are taking place and being conducted by the microorganisms and viruses. Here's an example of how we can, how we have a hydrothermal vent and how we can insert a sampling equipment right into the fluids. We also conduct, as I mentioned, undertake field work with submarines. So this is the submarine Alvin. So this is not a robot. So this has a pilot and 2 scientists who can go down. There's a really nice image of a technician who is just checking after the submarine is lowered into the water. The technician is making sure, is just checking with the pilot to make sure everything is okay, asking if no water is coming into the submarine, and if everything's okay, then the submarine goes down into the ocean. So Alvin Alvin has 5 different windows, so we can look outside the submarine with 5 windows. As you can see, these windows just look like eyes. Even though this is an instrument that is a multi-million-dollar instrument, you can see that we use scientists use cheap equipment. These are milk crates, plastic milk crates, because they are very useful. They never compress. Anything that's made of softer material will compress, whereas milk crates are great for sampling in the deep ocean. They can take pressure very well. We've also used another ROV known as Sebastian. This is similar to Jason. Here's an instrument known as an elevator. So this is essentially just a platform where you can keep samples or other equipment. So this is lowered and kept close to a hydrothermal vent and just used for storage. Here is the workhouse of the ocean. So this is known as a CTD sampler or rosette. So these are just water bottles. You drop them by the side of the ocean, and then you can collect water. And now I'll show you some fun and, let's say, not so fun images. So here is a deep sea hydrothermal vent crab. Here are images of tube worms from hydrothermal vents. These tube worms all contain symbiotic bacteria that produce energy for the tube worms. The tube worm has no guts, nothing, only symbiotic bacteria, and the bacteria are using hydrogen sulfide. So sulfur compounds for energy. That energy is then transferred to the tube worm. And these tube worms are some of the fastest growing organisms on the planet. Here is an octopus that, this picture got cut off, but it's trying to attack a lobster. This is our favorite pastime on the ship, table tennis or ping pong. You can imagine as the ship is moving, playing ping pong can be a lot of fun. Here are some more interesting images. Again, an octopus going after a lobster. Here's a squid carrying eggs. And finally, a giant Pacific octopus. This is a really beautiful image where we could see the reflection of the submarine in the eyes of the octopus. And then, when I showed you all of these beautiful images, but then I have to show you this, where the bottom of the ocean is not as pristine as we would imagine it to be. Plastic pollution specifically has touched every part of Earth today. So when we go down, for the most part, it's a beautiful, beautiful environment. And then we see stuff like this, where there's just plastic all over. There are balloons we've seen, we can see, I mean, plastic degrades very slowly. If you can see, you can read everything over here. So you know exactly where this was manufactured. These birthday balloons are out here. And then different kinds of organisms can make homes in these. So for example, if you see, this is actually the tentacles of an octopus. So an octopus has made its home inside all of this plastic. So I just wanted to illustrate, how pollution affects even the most remote parts of Earth. And then finally, here's a fun image of a hydrothermal vent. So there's a beautiful hydrothermal vent image where you have the liquids gushing out, and all the smoke that you see typically in hydrothermal vents, the black color are iron sulfides. So again, sulfur compounds. And here's a face that we observed in a hydrothermal, in amongst these fluids. When you're on the ship for like a week or say a month to 2 months, you start looking through images and you can find all sorts of interesting patterns. Now, before I finish, I'll just quickly give you a sense of what it is that we do at these hydrothermal vents. And when you think of, say, sulfur processes, at hydrothermal vents, we in the past had discovered novel viruses that possessed genes for sulfur oxidation. So we discovered, these DSR genes, which are important for the oxidation of elemental sulfur to sulfite. And now we've begun to look at the diversity of these sulfur genes. Initially, we had only discovered the DSR-A and DSR-C genes, but we have a paper that is very close to coming out, where we've expanded this to find these genes in a variety of environments, even outside hydrothermal vents, as well as identify genes associated with thiosulfate oxidation as well on viruses. Our working model associated with virus-enhanced sulfur oxidation is very analogous to what I showed you with cysteine earlier. The idea being that you have a bacteria. So in the deep sea and in other environments, you have bacteria that can oxidize sulfur and form intracellular sulfur globules. So they are storing intracellular sulfur. This is analogous to bacteria having a lot of intracellular cysteine. When you have a phage come in and infect this bacterium, these viruses would express sulfur oxidation genes like the ones that I just showed. The bacteria would respond by upregulating their sulfur oxidation metabolism and utilizing the stored sulfur globules. The viruses would steal this energy and replicate inside of the bacteria. And eventually, we have a lot of viruses here, and bacteria are consuming the intracellular sulfur and will eventually die. So on the basis of this, we have what we've proposed as short circuiting of the traditional sulfur food web. So if you're wondering what this means, this is the idea that there is a very traditional food web. You have viruses that infect autotrophs. There is something known as a viral shunt, where the lysed organic matter. So the organic matter that comes from microorganisms can be taken up by autotrophs or by heterotrophs. And then you have this cycle that goes on to higher trophic levels. So there's a very traditional food web. And what we have sort of come up with is a microbial sulfur loop. The idea that there is a sulfur associated food web as well. And these viruses are essentially short-circuiting the microbial sulfur loop. So essentially you have these reduced sulfur pools. So you have different sulfur compounds. There is microbially mediated processes that generate energy, cell growth. So this is your traditional sulfur loop. But when you have viruses coming in, there is energy that's produced, but all of this goes into virion production and lysis of host cells. This part is sort of skipped. So that's the idea behind short circuiting of the microbial sulfur loop. Now, to summarize what I told you today, I showed you details associated with 2 different softwares, but in general, we have a lot of excellent bioinformatic tools that can enable us to characterize viruses from microbiome data. I showed you that viral metabolic genes are involved in a variety of sulfur-associated processes, primarily sulfur assimilation and sulfide production from cysteine. I showed you that hydrogen sulfide can increase viral fitness. Viral dark matter, including viruses and metabolic genes, can serve as an indicator of disease states, such as in Crohn's disease. And finally, I showed you that viruses are important members of marine microbiomes that can drive sulfur transformations. So with that, I will acknowledge my laboratory. So much of the work I showed you today was the work that was driven by my PhD students and my postdocs. I'd like to thank my funding agencies, USDA, NSF, Schmidt Ocean Institute, WRF, and JGI. And much of the work I showed you would not be possible without a lot of our collaborators in human microbiomes, freshwater microbiology, as well as deep sea and hydrothermal research. So with that, I will take any questions. Here's my email and my Twitter links as well. So if you have any questions that I'm unable to answer today, or if you do not ask today but want to get in touch with me, feel free to get in touch with me as well. Thank you again.
Thank you very much, Dr. Karthik. It was amazing to have those images this afternoon here in Brazil. So before we start, because we got a lot of questions from the public, but before we start, are you going personally to these trips to get the samples at the sea?
Yes. So in the past, I personally have gone. So the most recent times I went was in 2018 and 2019. I just had my first child. My son is now 6 months old. So we'll see if I can go in the near future or not.
You cannot. I'm telling from in behalf of your wife, you cannot anymore, no more.
Yeah, but we are funded to go again in 2022. So hopefully we will get to go again using a submarine in 2022.
Is that boring as it seems or not?
So boring?
No, it doesn't seem boring. I'm kidding. I'm kidding.
Now it is quite interesting. You know, I can sort of just give a very brief description of the submarine, because when you go into the submarine, it takes about 1 hour to go down and 1 hour to come up. And the first 10 minutes is super exciting. And then after that, there is no light. And then you see all of the bioluminescence. And the bioluminescence is beautiful.
It's amazing. It's amazing.
But after that, the bioluminescence disappears and there is nothing. So for half an hour, there is absolutely nothing. It is very boring at that point of time. That point of time, you just want to get to the bottom. You just want to get to the hydrothermal wings, but it takes a lot of time. Yeah, those are very long 30 minutes. Yes.
Okay. I'm kidding. Of course, it's not boring. It's scaring for me, at least, but it's not boring at all. So we have some questions. We want to start, Krieger?
Yeah, just want to add a comment, Karthik. Thank you so much for the nice presentation, and thank you so much for our didactic presentation about IREP and use sequences to measure activity of virus in this case, but you can use for other bugs as well. So I think the students really appreciate your very nice explanation about using sequencing data to measure activity. So I'm sure they appreciate that. And yes, we have several questions here. So try to go. Actually, one of them I just right in front of me. People are asking, can you identify new virus sequencings without databases bias? So can you see fundamental new sequences that can associate with virus, taking into account that they're using different databases to identify them?
So the answer is, to a large degree, yes. And the reason is that when we use the databases, it is not to learn. So initially, when we're using the databases and we are using machine learning models, it is not to identify what a virus looks like, but it is to identify what a virus does not look like. So you assume that you know what bacteria, archaea, plasmids, and eukaryotes look like, and you remove those sequences first. And then whatever sequences are left, it is likely that a lot of them will be viruses. So you take those sequences and then you identify viruses to the best of your ability. So that's the approach we take.
Okay, so a follow-up question that is pertaining to this point is how you using the same approach, can separate, identify archaeal viruses, because they look quite different, right? The genomes are quite different. So how can separate them from other viruses?
Right. So first, when we we have identified a lot of the sequences that we think belong to viruses, and at that point of time, what we do is we begin to look at specific characteristics. With every virus genome, be it an archaeal genome or a bacterial archaeal virus genome or a bacterial virus genome, there are certain characteristics. Viral genomes are often segmented, so you will see there are structural proteins. There are metabolic proteins, and then there are replication proteins. So it just depends. So with archaea, you're absolutely right that often archaeal viruses look very different. But at the end of the day, all viruses have to undergo similar processes. They all have to replicate. They all need a polymerase. They all need, most of them, let's say, have terminases. There are other structural proteins that they need. So you can use patterns, but to be completely honest, if there is a very, very novel virus that no one has ever seen before, then it is possible that we might miss it. So there is a little bit of dependence in what you can identify, but you're absolutely right. So, what can happen is our software might call something a virus. And when you look at the annotation, or when you look at the proteins, it might just be protein 1 is unknown, protein 2 is unknown, protein 3, all the proteins are unknown. So the question is, you can still identify it as a virus, but you will not be able to identify it as a virus that infects archaea, because there's no way to tell that. Similarly, you won't be able to identify any other proteins as capable of anything.
Okay. So we have a question here. If you are interested in other biogeochemical cycles other than sulfur, are you interested in studying this?
Yes. So often, what happens as faculty is, our students and postdocs drive a lot of our research. So it's, partly their interests that really have driven where I've gone. So my primary interests are in sulfur. But I am exploring more. So my lab is exploring a lot of nitrogen transformations as well today, because in the oceans, in the freshwater lakes specifically, nitrogen is also very important. It can cause eutrophication as well. So I have a PhD student who is looking at the interplay between nitrogen, phosphorus, and sulfur, and how that can, cause transform lake health. So yes, we are doing others, but it really depends on, how different lab members drive our research.
Another question from a student of ours, Dilep Kumar. He is asking how machine learning detects or identify the interaction between microbiome and virus in the software you mentioned. Because you get some genes from virus and some genes from the bacteria. How can you detect this in the database?
So as far as the machine learning model goes, what the model is trained on is a few different databases. So it's trained on KEGG, PFAM, and something known as VOG. The idea is each of these databases has a different ratio of microbial and viral proteins. So like PFAM, for example, is rich in microbial proteins. KEGG is rich in microbial proteins. VOG is rich in viral proteins. So we use a combination of 27 different parameters to provide something known as a V-score to every protein. Now, what is a V-score? V-score is a measure of how virus-like a protein is. So in a given genome fragment, we annotate the proteins, and every protein is given a score, a V-score. And that V-score, eventually, whatever the V-score of the entire genome fragment looks like, determines whether it's likely to be a virus or not. And this is also what helps us identify a prophage. Because if you have a prophage, then the host genes 1, 2, 3, 4 may not look like a virus at all, and then all of a sudden you have genes that look like a virus. So that's sort of how we identify the virus. But when it comes to microvirus interactions, there's only a few of them that we can identify today. And say what I did speak about. So we can identify metabolic reactions. So whenever you have metabolic pathways or genes that are present on a viral genome, we can identify those, we can identify who they are similar to. So that's a measure of link between a virus and a host. So the virus is likely to have stolen those genes from the host. But we can't, so our software right now doesn't explicitly identify hosts for viruses. We are working on something different that can do it. It's a little more complicated. There are some easy ways to link viruses to hosts, but they only work for like 1 to 10% of all viruses. So then what happens to the remaining 90%? So we are working on newer machine learning approaches that will help us identify more viruses as well as say link viruses to their hosts. And unfortunately, yeah, they're not very complete right now.
Go, Krieger.
The next, just a second, because I lost
Paula
my connection.
Esteban.
Yeah. So, Karthik, you are just going viral right now. A person from University of Oslo is asking a question.
No, not really. We are doing both. I like the connotation there. I hope this talk is going viral. I would like that. But I think our lab, we are trying to do both microbial and viral work. It's just that right now a lot of our questions are heavily virus focused. But I have, it just really depends. Yeah.
Yeah, that was my question. So the real question from Paula from University of Oslo is, these tools can be used for large data sets, like 900 human samples. How the output will look like? So probably is a user already.
Yeah, so you can certainly use these on large data sets. It just sort of depends on how large the data sets are. I don't think the data set size is a limitation. We routinely, so I haven't, I mean, my lab typically uses, for human microbiome samples, we've used them on 200, 300 samples. That is not a problem. Most recently, we used a couple of hundreds, about 500 samples. That is never a problem. I don't see why it would be a problem for 900 samples. You might generate a lot of data, but it really depends on your questions. Are you trying to look at the differences between all 900, or can you pool all of them together? It really depends. Speed can depend really. Like for Propagate, because it uses read mapping, it is a little slower than Vibrant. You have to input read data sets as well, and mapping the read data sets is a limitation. But if you coded it and you just let it run for a few days, it'll be fine. So I don't think there is any limitation. You can use it in any way you want. And if you have any issues, please send us a message by email or on GitHub or anywhere, and we'll be very happy to help you out.
And actually, the next question is very technical too. So we skip it and ask the student to send direct to you. Okay. So the next one is from Giovanna Carvalho from here. And she said, and she is asking, it's known that computer algorithms, researchers are able to analyze data from the microbiome and gain new insights. But it's not clear to me how these methods can generate these insights for solving problems like treating diseases. She's asking how to apply this information, these insights for like treating diseases, some more practical results.
Right. So I think that's an excellent question. Right now, where bioinformatics and computational biology of the microbiome is I don't think it should be used as a tool to enable treatment of diseases. But what it can be used for, and I think where we're going towards step one, is helping to identify diseases. So I think that's a more important target that we are trying to achieve first.
And be also predictable.
Yes, so predictions and automation and being able to use it rapidly on hundreds to thousands of samples. So that's sort of the goal right now for computational biology tools. As far as say treatment goes, I think what it can hint towards the role of certain organisms or certain proteins, which would then have to be followed up followed up, with experiments. So for example, today we know that coronavirus is an important human pathogen. So what we can do is use computational biology tools, maybe even Vibrant, to go in and look at, let's say, if we are interrogating wastewater samples or other samples, we can see if there are novel coronaviruses in those samples. So that is where, so prediction or identification is the near-term goals. So for example, I know that even today, when people are looking at infections, there's still a lot of people are trying to do cultivation. So cultivate a pathogen and see if it grew, or a particular bacterium, did it grow from infections? Whereas you could easily switch to sequencing and identification using computational biology tools, which would be much quicker. So that's, I think, the goal. Treatment is an eventual goal, but I think it is very much into the future.
No, I'm an immunologist, and the microbiome just explodes in immunology. And you see microbiome of healthy people and people with autoimmune disease and things like that. They also propose some treatments like feces feeding of people, just trying to get a new homeostasis of this microbiome. So I think maybe we have to change minds and do not think in a specific medicine for a specific site. We have to change globally because we can maybe induct and manipulate this microbiome in the direction to health, right, instead of the disease. That's my point. It's more philosophical, I think, but I think we can achieve this in the future.
Right. No, you're absolutely right. So fecal microbiota transplant is probably one of the best case scenarios of how microbiome science is driven a very specific treatment. But yeah, in most cases, we are exploring how we can use probiotics and help modulate the gut microbiota. How maybe we can use phage therapy because of so much antibiotic resistance.
It's actually an old idea. I remember in the 70s of the last century, someone just having a new medicine, polyphage, for treating throat ache. It was a virus that enters in streptococcus, for example. So it's very interesting, very interesting. But we have another question, lots of questions. Go, Krieg.
The question is from Bruno. I was wondering if you have any data or can talk about the giant viruses that are found in the deep sea environments, especially regarding the presence of unique and uncommon metabolism-associated viral genes.
So that's an excellent question. So these are giant viruses that typically infect protists or algae. Unfortunately, our lab does not work on the giant viruses. So we do get them from marine systems, but we don't explicitly work on giant viruses, primarily because for the most part, we are only interested in phage, so viruses infecting bacteria. They're very interesting, but I just think bacteria, viruses that infect bacteria are cooler.
Another question from Sverdrup. I think that's the name. Could you talk about how water samples collected by the ROV are prepared for sequence and what sequencing platform is used?
So typically, once we get filtered, so when we have filters from from say the ocean systems, we ship them back to the lab on dry ice. Once they're in the lab, we will extract DNA from these samples. We can use kits, so we have used your traditional Mobio or QIAGEN kits, but sometimes they don't work as well. So when for deep sea samples that are very high in sulfur and metal concentrations, we prefer to use a standard phenol chloroform extraction for both DNA and RNA. After which we separate those two fractions and then sequence both the DNA and the RNA. For now, we have been using Illumina. We have used, Illumina all the way from Illumina 2000 to 4000. Now, we are using Illumina NovaSeq. But for the most part, my lab's, been focused on short read sequencing. But we have just started experimenting with nanopore and long read sequencing as well. So essentially combining the two, but we will see how that goes. But I would say our bread and butter is short read Illumina sequencing.
Okay.
The next one is from the lab, another one from him. Thanks for showing the wonderful video, Professor. I'm just curious about to ask about the collection of samples from hydrothermal vents. Can you share also some interesting things, any difficult you faced during these expeditions?
Yeah, well, difficulties. So we try and use in situ sampling. So, we will often include RNA later so that we can preserve the samples in situ, or we will filter the samples and then put RNA later on it in situ. Often that is not the case. That may not be possible. So if that is not possible, then we cannot do any RNA sequencing. So no transcriptomics, only DNA samples. So that's something. Often some of the challenges are not technical like this in the oceans. When you're doing field work in general, often the biggest challenges can be unpredictability. So you're on a ship, suddenly there is bad weather, and they will not launch the submarine for 4 days. But your cruise was only for 20 days. So if you lose 4 days, you lost 20% of your scientific experiment. So then you have to immediately come up with alternate ideas. There can also be equipment failure. So when you're down there with a submarine, and you have, let's say, 10 instruments in front of you, and you have a submarine arm that is not your hand or your fingers, right? So the submarine arm is not, it doesn't have that dexterity that your fingers or your hand has. So you might knock over something and then all of a sudden something doesn't work. So we have, been in situations where you lose hydraulic pressure or something, so you can't collect samples. So then you just give up your experiment for that particular dive. So unpredictability and accepting that is sort of the most difficult challenge, I would say, with field work in general.
The problem is because you are under the sea, not in the bench, because it happens in the bench too. So
Yep.
That's the problem for me. I'm scared. Okay, so Ícaro Castro is asking you, thank you, Professor, for such a nice talk. Which other disease are you interested? I think he's talking about this intestinal disease that you worked with, and if you have worked with another one or.
Yeah, so we have quite a few collaborations here in the med school, as well as with the Marshfield Clinic Research Institute, which is also in Wisconsin. So in collaboration with other scientists, we are also studying viruses that are in gut microbiomes associated with stroke and multiple sclerosis. So multiple sclerosis is a project that where we have gut samples from patients, whereas with stroke, we have mice samples. So we can't collect samples from people who have had stroke. It's a very technical issue. So with mice, yeah, there are experiments that are being conducted, and then we try and study the role of viruses in these samples. Yes, so we do study other systems as well, but we primarily do it through collaboration.
Mm-hmm.
And the last one here is from Judemar Crispin. He is asking, resistant genes of pathogenic bacteria have already been identified?
In virus. Virus.
Yeah.
Yes, so there are a few genes that have been identified. There's a little bit of controversy with antibiotic resistance genes on viral genomes. So we have identified a few, I would say, but then there are other genes that often look like antibiotic resistance genes, but they don't have the activity associated with antibiotic resistance. So the answer is a little bit of a yes and a no there. So, I think there's no reason why viruses would not carry antibiotic resistance genes on their genomes. If they can carry metabolic genes, and be associated with horizontal gene transfer, why wouldn't they carry antibiotic resistant genes? So that's sort of where I look at it from an evolutionary perspective.
So the follow-up questions for this one, Karthik, I'm having here is that we understand your hypothesis about how virus influence the sulfur cycle, right? So it's pretty strong evidence for that. But what about all the other genes, right? These metabolic, auxiliary genes that you see, even photosynthesis genes that are carried by these phages in environmental, like marine environment, right? So there are so many different metabolic pathways that are carrying around by the virus and being transferred laterally. So is there evidence for that you're actually doing something, or just more a fortuitous, packing or release of the phages?
Right. So that's an excellent question. So there is evidence that so viruses have been known to carry genes associated with a number of pathways. So they carry genes associated with phosphorus starvation, as well as transformations, nitrogen transformations, carbon transformations, specifically photosynthesis, because that's the best known example. So you're absolutely right there. As far as the actual process that these auxiliary metabolic genes are associated with, photosynthesis is the best example, because there are very good model systems. So people have studied that the virus photosynthesis proteins, they produce transcripts, they produce proteins. There's very good evidence that the viral proteins have a role in photosynthesis. With the other processes, the evidence is often only from omics data. And the reason is that we just don't have model systems right now. Everyone's trying to generate these model systems, but it's, already we know that isolating bacteria is difficult, but isolating viruses is even more difficult, because you first need to isolate the bacterium, only then can you isolate the virus. Now, as an example, if I wanted to study a virus that contains a sulfur oxidation gene, there's a very small chance I'll be able to isolate the bacterium. And then there might be 100 different viruses that infect that bacterium, but only one of them will carry that sulfur oxidation gene. It might be very important in the environment, but trying to translate them in the lab is not always easy. Now we have a workaround, which is that we have some model systems. So we have a model system associated with cysteine degradation in the lab. So we have the host, we have the virus. In that particular scenario, we've done a variety of analyses. So we've done transcriptomics, qPCR, proteomics. So we know that the viral metabolic genes are producing proteins. But it's not always that straightforward. We're also conducting some experiments where we are trying to insert metabolic genes in an E. coli T7 system. So this is a T7 virus that infects E. coli. You insert the metabolic gene into it and see if that virus is producing the proteins, and it has a different impact on the E. coli. It's, that can be a little bit of a hit or a miss, but we are trying different approaches to test predictions, especially associated with metabolic genes.
And I think that we are running out of time, right?
Yes, and it was very nice. We are very grateful for your presence here with us this afternoon. It was very, very nice. I'm quite sure the students and the other people that were in the YouTube like it, and will go to Twitter to see the other videos, other nice videos. So thank you very much. It was very pleasant to be here.
Thank you very much, Karthik. It was very nice.
You.
I'm sure the students will go after you. Don't worry about it. If you receive tons of emails from Brazil, that's fine.
No, I'm looking forward to that. I would love that. So thank you again for the invitation to present. I really enjoyed myself and answering all of the questions after the lecture as well. This is very enjoyable.
Okay, thank you
so much. Thank you.
Thank you. Bye.
Bye.
Bye-bye.